Good morning students, I am Mayank Tiwari. Today we will solve test number 53 of the General Agriculture series. Let's start. Student question number 1. Transfer of young seedling into another bed or pan or tray to have fast or vigorous growth of seedling and minimizing transplanting time. I again repeat the question. Transfer of young seedling into another bed or pan or tray to have fast or vigorous growth of seedling and minimizing transplanting time. What is this known as? Option A, thinning. Option B, pricking. Option C, hardening. Option D, mulching. So student, this question is very easy. So student, in nursery there are few essential operation like thinning. It means removal of excess of seedling to facilitate better development and aeration. Pricking is transfer of young seedling into another bed or pan to have fast or vigorous growth and minimizing transplanting time. Hardening, nursery plant are very tender, and to avoid excess field mortality, irrigation in nursery is withheld about 7 to 10 days before transplanting so that they can tolerate to extreme growing condition. This is known as hardening, student. In hardening, we withheld the irrigation 7 to 10 days before transplanting. Mulching, it is a layer of farm waste or sawdust or ash or polythene material, stones, etc. applied on soil to conserve soil moisture. Student, in case of growing media, please remember some important growing media like sphagnum moss. It has a water retention capacity of 15 to 20%. It is a bog grass and it can hold as much as 10 to 20 times more water as compared to its weight. And student, the pH of sphagnum moss is 3.5 to 4. And student, perlite. Perlite is a grey white silicaceous material obtained after processing of crude ore, and it can hold 3 to 4 times more water than its weight. Its pH is 7 to 8. It doesn't contain any mineral nutrition. So please student, remember it. Perlite is used in combination with peat moss. It is a common rooting media for cutting. And student, vermiculite. It is a micaceous material obtained from mica ore, and it has got very high cation exchange capacity. And student, vermiculite also contains magnesium and potassium, which is important for growth of plant. Student, peat. Peat has a pH 3.2 to 4.5, and peat can hold 15 times more water as compared to its weight. And student, this sawdust is also a common rooting media for nursery, and it is a waste product from timber industry, and it can hold sufficient moisture and it retains sufficient moisture for a long time. So remember sawdust also. Student, question number 2 you see. Autonomic parthenocarpy is found in option A, pineapple, option B, black corianth, option C, Allahabad round guava, option D, all. You have to tell in which of the following fruits autonomic parthenocarpy is found. So student, parthenocarpy you all may be knowing. It is a formation of seedless fruit without the act of formation of seedless fruit without pollination and fertilization. It is of two types. Vegetative parthenocarpy and stimulative parthenocarpy. Vegetative parthenocarpy is also called autonomic parthenocarpy. It means ability to set up fruit without stimulus resulting from pollination. Example, banana, pineapple, some variety of fig and orange. Student, this variety is very important. Washington navel variety of orange has vegetative or autonomic parthenocarpy, and this stimulative parthenocarpy or autonomic parthenocarpy, it means seedless fruits are developed only in response to external stimulus to ovary. Example, black corianth variety and Allahabad round guava. These are stimulative or ationic parthenocarpy. And induced parthenocarpy, student, production of seedless fruit by treatment of flower with materials such as that pollen or pollen extract and other growth substances like IAA, IBA, IPA. This is induced parthenocarpy. There is one more type of parthenocarpy, student, please remember, stenospermocarpy. It is formation of seedless fruit in which pollination and fertilization take place but embryo get aborted. Please remember, student, in stenospermocarpy, pollination and fertilization take place but embryo get aborted, and the example of stenospermocarpy is Thompson seedless variety of grape. Student, question number 3 you see. Spices are generally perennial except, you have to tell which of the following is not a perennial spice. Option A, ginger, option B, coriander, option C, turmeric, option D, all. So student, classification of spices, you must remember some example. First, rhizome spices, example ginger, turmeric, garlic, these are rhizome spices. Then seed spices, coriander, nutmeg, fennel, cumin, fenugreek, and celery seed. These are seed spices. Bark spices, cinnamon. Fruit spices, chili, black pepper, cardamom, tamarind, vanilla. These are fruit spice. And leaf spices, bay leaf and curry leaf. Flower spices, saffron and clove and asafoetida. Asafoetida is also called hing, and clove is a unopened bud. Please remember it. And the student, saffron. Saffron is the costliest spice of the world, and it is also called red gold. It is grown in Kashmir region of India. So please remember saffron. Saffron belong to family Iridaceae. Please remember it. And also students, some tree spices you must remember: clove, nutmeg and cinnamon. These are tree spices. And the student, difference between spices and condiment you must know. Spices have pungency principle, that is they are meant for pungency, and condiment are meant from two word. Condi means season and ment means flavor. So they are used for seasoning purposes, and they add aroma and taste to any material. And spices are used in pieces except turmeric, which are used in pieces. And condiment, these are used as powder or seed. Most authentic difference between spices and condiment is origin. Spices are tropical in nature when it comes to origin, and the condiments are of temperate origin. And most of the spices are perennial except ginger and turmeric. So the correct answer to this question was ginger. And condiment are mostly annual except coriander. And in spices, localities of South India, and condiments, these are mostly North India confined. So student, please remember the differences also. Student, question number 4 you see, complementary gene interaction is also called. Option A, duplicate recessive interaction. Option B, recessive epistatus. Option C, amorph. Option D, hypomorph. You have to tell what is complementary gene interaction known as. So student, please remember, complementary gene interaction is also called duplicate recessive interaction, and the ratio obtained in complementary gene interaction is 9:7. In duplicate dominant interaction, 15:1 ratio is obtained. Supplementary gene interaction is also called recessive epistatus. The ratio obtained in supplementary gene interaction is 9:3:4. Student, this is damn important. Always asked one of the ratio they will ask you in any competitive exam, so please remember the ratios. Inhibitory gene interaction, the ratio is 13:3. Please remember it. Student, one of the term I gave was amorph. So what is amorph? Amorph is an active allele which acts as genetic block to normal biosynthesis. Please student, remember amorph. It is an active allele which acts as genetic block to normal biosynthesis. It is known as amorph. And this classification of allele is based on action. It has been given by Muller. And hypomorph is allele that function imperfectly in comparison to wild type allele. So student, please remember hypomorph also, allele that function imperfectly as compared to wild type allele. This is known as hypomorph. Another classification of allele is hypermorph, allele that produce excessive amount of product as compared to wild type allele. This is known as hypermorph. What is antimorph? Allele with an action opposite to wild type. This is antimorph. And one is student, neomorph, allele whose action differs qualitatively from that wild type allele. It is known as neomorph. Please student, remember these terms. These are often asked in BHU. So allele whose action differ quantitatively from wild type allele, it is known as neomorph. And what is isoallele, student? Both the allele are same, not able to differentiate phenotypically. Allele producing only very slight differences in phenotypic expression, making their identification bound to use of specific method. Student, isoallele cannot be distinguished phenotypically. A specific method you have to use for isoallele identification. So please remember isoallele. Student, also remember that the term allele was given by Johansen in 1909. And student, please also remember multiple allelism. It means more than two alleles are present, and multiple allele, student, are human blood group is an example of multiple allelism, and vestigial type of wing in Drosophila, these are also the example of multiple allelism. Please remember. And student, please also remember self incompatibility in Brassica. It is also example of multiple allelism. Please remember, self incompatibility in Brassica is also example of multiple allelism. Student, question number 5 you see, effect of pollen grain on embryo and endosperm is called. Option A, xenia. Option B, metaxenia. Option C, pleiotropy. Option D, cytoplasmic inheritance. So you have to remember what is the effect of pollen grain on embryo and endosperm known as. Student, first seek option number D, cytoplasmic inheritance. Please remember that cytoplasmic inheritance is also called extra chromosomal inheritance or maternal inheritance or extra nuclear inheritance or uniparental inheritance. These are all the example of names of cytoplasmic inheritance. Please remember. So some terms you have to remember in cytoplasmic inheritance. First term, plasma gene. Plasma gene are gene which are located outside nucleus and in cytoplasma. These are called plasma gene. What are mitochondrial gene? Which are located in DNA of mitochondria, and mitochondrial DNA plus cytoplasmic DNA. These are known as organelle. And what is plasmon? Sum total of gene present in cytoplasm of cell. These are called plasmon. What is called gemon? Sum total of all gene present in nucleus. These are called gemon. So please students remember these. What is xenia, student? Effect of pollen grain on embryo and endosperm. It is called xenia. Example of xenia is maize and maize coloration of seed. It is example of xenia. Metaxenia, effect of pollen grain other than an endosperm. It is called metaxenia. Example of metaxenia is in cucurbit. Skin color is affected by metaxenia, and in case of orange also metaxenia is found, and in case of cotton hair length also metaxenia is found. What is pleiotropy, student? Pleiotropy is one gene having multiple effect. Sometimes various characters are controlled by a single gene. It is called pleiotropy, and in pleiotropy recombinant are not possible. Example of pleiotropy is sorghum. In sorghum, length and width of leaf and length of panicle are controlled by the same gene. So please remember sorghum. And this pleiotropy is also found in case of pea, students. In case of pea, flower color, seed coat, color of leaf are controlled by the same gene. So pleiotropy is also found in case of pea. So student, that's all for today. Thank you.